with that, it's a real pleasure to introduce Siobhán O'Brien. Since 2021, she's been an assistant professor in the Department of Microbiology at Trinity College Dublin. She also received her undergraduate degree there. And in between, she has a long pedigree of impressive institutional and other affiliations, ETH in Zurich, the Wissenschaft Kolleg in Berlin, many other places. She was also, one of her papers was a finalist for the Williams Prize, prize for papers in evolutionary medicine. And her research focuses on the ecological and evolutionary dynamics of complex microbial communities. And so with that, I'll turn things over to you, Siobhán. Thanks for joining.
Thank you so much. Let me see now if I can. Okay, is that okay?
That's great.
That's brilliant. Okay, thank you so much for the introduction, and really appreciate being invited to speak to you all today. So my name is Siobhán, and I study conflict and cooperation and general social interactions in microbial communities. So I study microbial communities in ecological contexts like soil, but today I'm going to talk to you about the work that we've done on human microbiomes. So in the cystic fibrosis lung, but also in the gut microbiome as well. And so we're all on Zoom. So I'm just going to try and sort of invite you into where I am at the moment. So I'm in Dublin, here on the east coast of Ireland. So we have a population of just over 1 million, which is really big by Irish standards. I'm based here in Trinity College. And if you look down here on the right-hand side, this is the Department of Microbiology. So it's a really beautiful campus. You go through the main door and you come out to this lovely cricket pitch. And here we have the microbiology department. And this yellow building here is our student bar. So our lab here on this corner looks right over onto the PAV, or the beer garden in the PAV. And so we can see all of our students that should be in the lab having fun in the PAV instead. And my office that I'm sitting in right now is just over here. So Dublin's great, so lots of nice bars and lovely beaches. And so if anyone's interested in what I'm doing, I'm always interested to hear from sort of potential students or postdocs that might be interested in coming to join me. I should also say that it's 5 o'clock here and it's about 1 degree Celsius and it's dark outside. And everybody's really excited because there's a prospect of snow, which is very unusual here. So we will wait and see now if there's travel chaos on the way home. Okay, so as I said, I'm really interested in the social lives of microbes. So this really began during my master's project at Exeter. So this is way back over 10 years ago. And during this time, I was studying cooperation in these populations of white-browed sparrow weaver birds. So these live in the Kalahari in South Africa. And they form these social groups where they engage in lots of cooperative activities like nest building and vigilance against predators. And so I spent a couple of months doing field work out here, and I was really studying sort of the contribution to cooperation by different individuals. So why do some individuals cooperate more than others in this system? And so during this time, I was kind of reading literature to write up my thesis, and I came across this body of literature by Stu West, Ashley Griffin and colleagues at Oxford. And they were really trying to ask more or less the exact same questions that I was trying to ask in birds, but using a very different system. So using microbes instead to ask these kinds of questions. And I thought this was really, really amazing. So you could study populations that instead of consisting of 10 individuals, they consisted of millions of individuals in a test tube. You could have replicate populations where you can study them at the same time. And of course, you can evolve bacteria in sort of in real time. So you can evolve them over very short timescales. And so I decided to sort of venture into the lab for the very first time and do my PhD in microbiology. So really looking at the social interactions in microbes instead of birds. And that didn't go very well. So this was in the first week of my PhD, where I decided to autoclave a 96 well plate, which is made of plastic, and of course wrecked the entire autoclave. But thankfully, it was all uphill from there, and I really settled into life in the lab okay. So I'm really interested in social interactions now in microbial communities. And we know social interactions are really rife in microbial communities. So these bacteria don't exist in isolation. There are sort of negative interactions like competition between individual species for resources, and predation between bacteria and their microbial predators, and even bacteria and their phages. And bacteria also engage, of course, in positive interactions such as cooperation. So individuals can share public goods. So they produce public goods into the environment, and then they can take up public goods produced by others. And this is really what I'm going to be focusing on in this talk. So lots of different social interactions can occur in microbial communities. But I guess the question we're trying to ask in our lab is whether or not they actually matter. So we know that social interactions do occur, but do they actually matter for things like virulence, when we think about infections and the ability of a pathogen to cause disease? Do they matter for bioremediation, so the ability of microbial communities to break down heavy metals or detoxify heavy metals in soil? We also look at social interactions with regards to food security. So how social interactions in microbes actually promote the health and the growth of our food crops. And then finally, we look at social interactions and how they can drive antibiotic resistance. And so very, very similar concepts, but in very, very different systems. And so to try and sort of get at these questions, we tend to use systems that are kind of trying to bridge the gap between these really simplified environments and then analysis of natural populations. So we do some experiments in these simplified test tube environments, but we also have these sort of semi-natural microcosms, so gut microcosms, or cystic fibrosis, artificial sputum microcosms. And we also do experiments in soil in the lab as well. And so today what I'm going to focus on are the gut microbiome, social interactions, and social interactions in the context of respiratory infections. So really focusing on these human microbiomes today. So in the first part of the talk, I'm going to talk to you about social interactions and the impact they can have on virulence in Pseudomonas aeruginosa infections of the cystic fibrosis lung. And vice versa. So how does Pseudomonas aeruginosa virulence actually drive social interactions in the microbiota as well? And so this project has really gone on for maybe 10 years now, and it started during my PhD, and we've still continued to develop it up until now. So it's a huge collaboration between myself, Mike, who was my postdoc supervisor while I was at York, he's now at Manchester, Angus, who is my PhD supervisor at Exeter, Sive, who's my PhD student here at Trinity, and Joe, who's a long-term collaborator in CF microbiology, who's based at Liverpool. So the social sort of behavior we're going to focus on is the production of siderophores by Pseudomonas aeruginosa. So Pseudomonas will often find itself in sort of iron-limited environments. So iron might be bound to proteins in the body, for example, or minerals in a rock. And so to overcome this, it's able to produce these iron chelating siderophores, so depicted here by this sort of green, very crude shape. So this bacterium is secreting the siderophores. The siderophores can then bind iron that's sort of otherwise unavailable in the environment. So it's bound to a protein in the Fe3+ form. So it can bind to iron, and then it can take this iron complex back up into the cell. And it has to have a species-specific siderophore receptor to do that. And this behavior is very interesting from a cooperation point of view, because it's not just the individual who produces a siderophore who can actually get the benefits. So any individual, providing it has the correct siderophore receptor, can evolve or sort of arrive into the population, use the siderophores produced by others to access iron, and then not actually produce the siderophore itself. And so you can kind of see how this is a public good. So individuals produce the siderophore into the environment, and then individual cells can essentially take the siderophores from the pot. And so like all public goods, this is going to be vulnerable to cheats because these yellow individuals here who don't produce siderophores don't pay a cost of production, but can actually still get the benefits, providing there's enough siderophores in the environment for them to take up. So they won't pay any cost of production, but they'll still get the benefits. And as a result, the non-producers should quickly spread through a population of producers. And so you can show this quite easily in the lab. So you can essentially initiate some populations with just a clone of a producing cell, so Pseudomonas aeruginosa in this case. So you can have 6 different replicate populations, and you can evolve them in the lab under iron limited conditions, and then every couple of days quantify the evolution of de novo cheats. So these are non-producers that aren't producing siderophores. And you can see that after 25 days, really these populations then are mainly composed of non-producers. And if you do this in iron-rich environment, you don't find this rapid increase in non-producers because the siderophore production is essentially turned off in an iron-limited environment, or an iron-rich environment, sorry. Okay, so cheats evolve really rapidly. So siderophores are really important for virulence, so they allow bacterial pathogens to establish an infection. And so if you infect a little wax moth larva here with a siderophore cooperator, so a siderophore producing strain of Pseudomonas aeruginosa, or a cheat, so a non-producing strain of Pseudomonas, you can see that the cooperator can actually kill the host faster than the cheat can. So they really are quite important for virulence as well. So these non-producing cheats are obviously quite bad from a bacterial population perspective, but actually if you're a host and you're infected by Pseudomonas aeruginosa, they could be quite a good thing because they are reducing virulence of your pathogen that you're infected by. And so we wondered whether we could manipulate the host environment in order to artificially enhance the spread or the evolution of cheats in the host. So we know that de novo evolution is ultimately limited by mutation rate. So mutation rate is a good predictor of how adaptable a pathogen is going to be. So in general, when we have a much faster mutation rate, we tend to see faster adaptation to a novel environment. And one way you can artificially increase mutation rate is by exposing bacteria to temperate phages. So these are phages that essentially can integrate their genomic material into the genome of the host. And in doing so, they cause mutation. So they can integrate into the host genes and therefore cause mutations and structural rearrangements in those genes. So they essentially act as a hypermutator or accelerate the rate at which these novel mutations appear. And so we previously showed that if you evolve Pseudomonas aeruginosa in artificial sputum with and without temperate phages, the presence of these temperate phages will actually drive adaptive evolution to this biofilm lifestyle. And that's because it's essentially providing a novel supply of mutations for more rapid adaptation. And so we hypothesized that actually temperate phages should also drive selection for cheats in an environment that's iron limited, because cheats are going to be favored in this environment. So we had this experimental system where we had Pseudomonas aeruginosa and we had a temperate phage known as 5,4. So Pseudomonas aeruginosa is a pathogen of the cystic fibrosis lung. We use PaO1 in this experiment, although it's not a CF isolate, and that's because a lot of CF isolates already have temperate phages in their genome. PaO1 doesn't. And that's why we used it for this particular experiment. The temperate phage we were using, called 5,4, doesn't actually carry any beneficial mutations per se. So lots of temperate phages actually have beneficial mutations in their genome, and that can be a reason why they are retained in the host genome. So it doesn't carry any beneficial mutations per se, and it integrates more or less randomly throughout the genome. So it doesn't seem to have any affinity for particular hotspots within the genome. And it's clinically significant. So this phage was isolated from a patient with cystic fibrosis. And so what we monitored was the frequency of these non-producing cheats over the course of 30 days of evolution. So we had an iron-rich condition here on the left-hand side, and we had an iron-limited condition here on the right-hand side. And so all of these different lines correspond to different replicate evolving populations. So what you'll see is in iron-rich environments, we don't really see the evolution of cheats. And that makes sense because siderophore production is not expressed in that environment. So there's essentially nothing to cheat off of. But if you look at iron-limited environments, we see that cheats evolve quite quickly in an iron-limited environment without phage. So you get some cheats evolving. They're not reaching very high frequencies, but they are evolving. And they are sometimes spreading to quite sizable frequencies. I should say that this is a different bacteria medium sort of combination that I showed you in the previous slide, and that's why I think we don't see as much cheating going on here as we did previously. But the important thing is that if you perform this experiment under iron-limited conditions with our temperate phage, you can quickly see rapid selection for cheating. So you get quick, really rapid loss of siderophore production in this treatment, and you don't find this when the environment is iron-rich. So this really suggests that these phages are essentially accelerating the selection for cheating in this pathogen. And so we also did some whole genome sequencing of our evolved populations, and we basically found that the reason these phages are driving selection for cheats is that they're integrating into genes that are associated with siderophore production directly. So a lot of these regulators like GAC-S, for instance, tend to be disrupted via phage integration events. So we can often see that the phage material is directly sitting in those genes and therefore making them dysfunctional. Okay, so clearly siderophore cheats can evolve quite quickly in this artificial environment, and temperate phages can accelerate the evolution of cheats, potentially reducing the virulence of an infection. But of course, cooperators are also going to be evolving as well. And so the next question we wanted to ask was, can cooperators actually adapt to a high frequency of cheats? And if so, how could they do this? And so this really stemmed from conversations with a behavioral ecologist, Mike Kant, down at the University of Exeter. And societies have loads of different ways of dealing with cheats. So if we look at sort of honeybees, we can see this policing activity. We can see reciprocity is a huge way of constraining the evolution or the appearance of cheats. And in microbes, we also see mechanisms that constrain cheating. So for example, in yeast, they have this flow1 gene that only allows them to flocculate with other bearers of that flow1 gene. There's also D-discoidium, which forms these spores. So essentially have a multicellular spore held on top of this stalk cell. And so individuals will basically cheat and become overrepresented in the spore and avoid turning into these non-reproductive stalk cells. And there's ways of policing this. So for example, if you turn into, if you're overrepresented in the stalk, sometimes the spores are much smaller and actually less viable. And you also have things like green beards. So green beards, for example, this flow1 gene has been an example of a green beard in bacteria, potentially, but it's a way in which individuals can link the cooperative behavior with some phenotypic attribute. So individuals only cooperate with other carriers of that gene. So there's lots of ways societies can sort of deal with cheats, and what we wanted to see was in this siderophore system, could our siderophore cooperators actually evolve to be able to mitigate this negative impact of cheating? And so we set up another experimental evolution experiment where we evolved populations consisting of 100% cooperators or siderophore producers, and another condition where cooperators were kept at just 10% of the population. And 90% of the population consisted of these non-producing siderophore mutants, so non-producing cheats. So we used PA01 again as our siderophore producing strain, Pseudomonas aeruginosa, and we had an isogenic cheat that had the main siderophore pyoverdin and a secondary siderophore pyocalin knocked out. So it really wasn't producing any siderophores. And so we evolved these different populations for about 200 generations. And at the end of the experiment, we isolated the cooperators only. So these cheats were not evolving, only the cooperators were allowed to evolve. And we performed fitness assays to see whether or not the cooperators had indeed adapted to the presence of cheats, and also phenotypic assays, so really to identify how they had adapted to cheats. And so we measured siderophore production for all of our isolates evolving without cheats. So these are just the cooperator only populations, and our isolates evolving with cheats. And we compare them to our ancestral strain. And so what we found is that the population that had evolved in the presence of cheats had actually reduced its own production of siderophores. And we saw no difference between the populations that had evolved in the absence of cheats and the ancestor. And this was really interesting to us, mainly because we'd kind of anticipated that the opposite would happen. So our prediction was that actually cooperators evolving in the presence of cheats should probably be under selection to actually increase siderophore production, because there's so many cheats in the environment that there's not enough siderophores to go around. But we found the opposite. And it was also quite interesting because when we grew up these populations alone, just in an iron-limited environment, there didn't seem to be any cost associated with losing siderophore production. So these isolates that had really way lower siderophore production just grew just as well as our ancestral populations and the non-cheat evolved populations in an iron-limited environment. So they were somehow able to access iron, even though they had evolved to produce far fewer siderophores. And so we kind of looked at these colonies, and we could see differences in the colonies that came from our cheat evolved populations and our control populations. So in our cheat adapted populations, most of the colonies were these kind of novel morphotypes. And so they were much smaller, had a slightly raised surface and this sort of wrinkly edge. And when we grew them up, we could see that they were phenotypically quite different as well. So on the right-hand side, we can see these three tubes. They are the novel morphotypes grown up in LB medium. And on the left side, we have three tubes, and these are the smooth morphotypes in LB. This blue color is really synonymous with the antimicrobial piocinin. So we decided to measure piocinin, and lo and behold, the populations that had evolved with cheats tended to produce much more piocinin than those that had evolved without cheats. And we did some sequencing and found that yes, in these novel morphotypes, there did seem to be mutations in genes associated with piocinin production. So it did look like piocinin was somehow being upregulated in the populations that had evolved with cheats and not in our control populations. So why was this? So piocinin is a really harmful toxin. It can really damage host cells, and it's a potent antimicrobial agent that can reduce community diversity. But it's not just a toxin, so it's also a powerful reducing agent. And what this might mean is that it's able to reduce iron from the Fe3 plus form, so this unavailable form, into the Fe2 plus form. And the Fe2 plus or ferrous form of iron is able to just diffuse directly into bacterial cells. And so we hypothesized that actually these cooperators that had reduced siderophore production under selection from high cheat loads had instead upregulated piocinin production to try and get at iron in a very different way. And so we were able to test this by exposing populations of just cooperators, so here in red, and cheats here in blue. And we tested the effect of increasing concentrations of piocinin on the fitness of these populations. So this is the fitness of each population relative to its own growth in the absence of pyocynin. And this is all under iron limited conditions. And so what we found was that if you add pyocynin to populations of cheats, we can see that it actually rescues the poor growth of cheats in iron limited environments. And so these cheats are not able to produce siderophores, but when you add pyocynin, it's actually able to liberate iron from the environment and therefore giving cheats an advantage. And we don't see as much of an increase in the cooperator populations. So this really shows what's going on in this experiment. Yes, cooperators could adapt to the presence of cheats. They did this by reducing their own cooperative output and instead switching to a toxin, pyocynin. And I think this really sort of demonstrates that when we're ever considering sort of a novel therapeutic, so for example, trying to increase selection for cheats in a real infection, really doing these sort of long-term evolution experiments can be really a powerful way to understand the repercussions of that treatment. And so it might seem like a really good idea to actually reduce siderophore production in an infection, but if cooperators are going to be under selection to actually counteract the loss of siderophore production, pyocynin could be really not good news from a patient's perspective. Okay, so that was kind of all done during my PhD, and after this I moved up to the University of York, where I was working in Mike Brockhurst's lab. And so it was at this time I started to work with these natural isolates of Pseudomonas aeruginosa. So everything I've showed you up to now has been done on PA01, or this classic sort of lab strain of Pseudomonas. But now I'm going to talk to you a little bit more about these clinical isolates. So what I want you to see here is that, first of all, you can see some of these plates here are really saturated with that blue color, and that's pyocynin again. So you can see that here, it's really flooding entire plates. So every single one of these plates contains a single isolate of Pseudomonas from a cystic fibrosis patient. And this all comes from a single patient. So we had individual patients, and we were able to isolate 40 colonies from each patient and analyze them for pyocynin production. And so you can see pyocynin production is quite high in some of these isolates, but I also want you to note that it's extremely varied. So within some isolates within this this patient had absolutely no pyocynin production, you can see here, and some have really, really high pyocynin production. So lots of variation there within a patient. And so we decided to quantify this. So we had 3 different patients, and from each patient we had these 40 Pseudomonas isolates. This is all isolated from a single time point in those patients, so just a snapshot within those patients. And so we measured pyocynin for all these isolates, and essentially in 2 cases we find this huge amount of variation within a patient. So we have essentially very low producers or in isolates that don't produce any pyocynin living alongside isolates that are really, really overproducing pyocynin. And so the question we're trying to ask now at Trinity is, what is the consequence of this intraspecific diversity within a patient for species interactions in the CF microbiota? And vice versa. So how do species interactions in CF depend on pyocynin or intraspecific diversity within one of those species? And so this is work being done by Siobhán, who's a PhD student here at Trinity. And I'm just going to show you some very preliminary data now on what she's done. She only started in September, but there's some interesting results so far nevertheless. So we decided to focus on pyocynin production, first of all, because we know it's quite a toxic antimicrobial. And so we had data on the pyocynin production from all of these different isolates. So these are all 40 isolates from a single patient sample. This was CF03. And so we first just performed really simple growth curves for all of these different isolates and find that actually pyocynin production is, probably not surprisingly, quite costly when it's not required. So if you have isolates that are producing very little piocinin, the productivity of those isolates tends to be much higher than high piocinin producers. But if you now expose all of those isolates to the supernatant of another species commonly found in cystic fibrosis lungs, Staphylococcus aureus, what we find is that this intraspecific variation really translates into the nature of interspecific interactions between these two species. So the Staph aureus supernatant tends to actually increase the productivity of Pseudomonas that's avirulent or not producing much piocinin. But we look at Pseudomonas piocinin, high piocinin producers, we don't find any effect of Staphylococcus aureus supernatant. And I should also say this is really at odds with what we expect to see when we think about PA01. So PA01 is the Pseudomonas lab strain. And if you expose Staph supernatant to it, it really diminishes the productivity of that strain. So really, when we looked at these natural isolates, we don't see that inhibitory effect of Staphylococcus aureus, at least with supernatant experiments. But of course, these are all clinical isolates. So they're not only, they don't only vary with respect to piocinin, they vary with respect to lots of other types of secretions. And actually, we found the exact same effect for pioverdin, so a siderophore Pseudomonas aeruginosa. So if you measure pioverdin, and again, we look at the effect of Staph aureus supernatant on different levels of pioverdin production, we can see the same effect. So individuals that produce very little pioverdin tend to benefit in the presence of Staph, and vice versa for high producers, and lactate protease as well. And these are all secretions involved in virulence. And so essentially what you end up seeing is that the outcome of these interspecific interactions, so whether they are positive interactions or negative interactions or neutral interactions, essentially depends on what lineage these isolates come from within a patient. So this diagram is essentially all of these 40 isolates, and we have a phylogenetic tree for these 40 isolates. So they generally split into two different lineages. We have lineage A up here that consists of these avirulent, low piocinin producing subtypes. And we have lineage B down here that consists of these high virulent subtypes. And remember, this is all a snapshot of a single individual at a single time point. And what we find is that for lineage A, they tend to increase productivity in the presence of Staph, and lineage B seems to have no effect when it's faced with Staph. So really, what I've showed to you in the first part of this talk is that, yes, siderophore production is this public good that's vulnerable to invasion by non-producers. Cooperators can in turn adapt by upregulating piocinin, which is a toxin. And of course, piocinin and all of these secretions by Pseudomonas aeruginosa can actually be an important driver of interspecific interactions as well. Okay, so I'll move on now to the gut microbiome. So this was sort of a project that I did while I was at ETH Zurich in Alex Hall's lab. So he's got a really nice anaerobic gut microcosm system that he works with over there, which I really wanted to try out. So this is like quite a nice fun project that we were able to do together with Michael Baumgartner as well. And so we were looking at social interactions, of course, that's kind of what I'm interested in. But in this context, we were looking at the gut microbiome and antimicrobial resistance. And so just to sort of reacquaint ourselves, I guess everyone here is familiar with antibiotic resistance, but we think about how antibiotic resistance evolves. It mainly is composed of two different phases. So if we imagine we have a population of susceptible cells, antimicrobial resistant individuals can arise in this population either through de novo mutations or through immigration of novel genetic material into the population. And so now we have a scenario where we've got a population consisting of maybe, a handful or one or two resistant individuals, but mostly susceptible phenotypes. And the second step is selection. So in order for resistance to actually spread through the population, it needs to be selected. If it's selected, then it should theoretically spread through that population. And from a clinical perspective, I guess what's quite relevant, is that this first step consists of a population that is actually quite treatable. So if this is a pathogen and you've got maybe some resistant cells in that population, but it's not completely consisting of resistant cells, you could probably still treat that pathogen, right? So you could treat it with antibiotics, kill enough of the population so that the immune system can then kick in and hopefully kill the rest. But once you've got selection for resistance, this then causes a population to evolve that is essentially not treatable. So if most bacteria in that population are resistant to antibiotics, it won't be treatable in the clinic. So we were really interested in the second part of the process. So how does selection operate on a population that already has a resistant strain in there to really drive the evolution of resistance, so we have a population that consists of solely resistant individuals. And so selection can be really predicted based on two really simple concepts, right? It's the benefit of resistance, so how good are those resistance genes at actually conferring resistance, and the cost of resistance, so how costly is resistance when it's not needed, so in the absence of the antibiotic. And of course, if the benefits outweigh the costs, then antibiotic resistance will ultimately be favoured and selected and predicted to spread through that population. So we hypothesized that actually social interactions in the gut microbiome could be a huge predictor of how fast resistance could spread in a community. So for this, we focused on ampicillin resistance in E. coli. So the mechanism of resistance to ampicillin is normally by beta-lactamases. So ampicillin is a beta-lactam antibiotic. So that's this toxic symbol here. And our bacteria is going to produce beta-lactamases in order to detoxify that antibiotic, and it detoxifies it outside the cell. So all the detoxification occurs extracellularly. And this is really similar to siderophores, right? So this essentially could be regarded as a public good, because other cells nearby could benefit from a reduction in toxicity without actually paying a cost. So you can probably see where I'm going with this now. Okay, so when we looked at the literature, there were two really conflicting hypotheses about what we should expect when we think about how social interactions should shape the evolution of resistance in this system. So the first that we saw was cross protection, and this was really, closer to our field in terms of what we would expect. So this is the idea that this is a public good. So we have resistant cell, it's producing these beta-lactamases, it's degrading antibiotics, and then these susceptible cells get a benefit because they're not actually producing these beta-lactamases, but they're still getting a benefit. So they're being protected by the presence of these resistant cells in the environment. But there was another line of research that predicted that that wouldn't necessarily be the case, and what we should see is actually competitive release. And what this means is that our producing cell essentially is producing beta-lactamases, and these susceptible cells don't experience a benefit fast enough in order to benefit from it, because the antibiotic just kills them essentially immediately. So the antibiotic is killing the susceptible cells, and this is freeing up resources for our resistant strain to then thrive. So competitive release doesn't predict cross protection occurring, it just predicts that these susceptible strains will immediately die, and then this resistant strain can use up resources and it will grow faster. And so you can probably see that these two hypotheses have very different predictions. So with cross protection, we predict that the susceptible cells should gain an advantage, similar to our non-producing cheats in the previous example. But competitive release suggests that our resistant strain should get an advantage because it's able to use resources from dead susceptible cells. So we wanted to sort of develop a system where we could potentially test these two ideas simultaneously. So we were using this E. coli ampicillin system, and we first wanted to test whether cross-protection could actually occur in this combination of E. coli and ampicillin context. So this is just done in simple lab media. And what we did was we were able to grow our resistant strain in green and our susceptible strain in yellow. We either grew those strains alone in the presence and absence of antibiotic, or we grew them together in the presence and absence of antibiotic. And the prediction was that the susceptible strain should do really badly when it's alone in the presence of antibiotic because it's being killed. But if you put that susceptible strain into co-culture, suddenly it should be able to be rescued by the resistant strain, okay? If cross-protection is occurring. God, I can't control it. So what we found is that if you grow our two strains alone, so in sort of isolation in the absence of antibiotic, our susceptible strain is growing really well, better than the resistant strain. So potentially there's some cost of resistance in the system. But of course, when you hit this then with ampicillin, our resistant strain, there's no effect on our resistant strain, but our susceptible strain is killed. So the growth of our susceptible strain is greatly reduced when you add ampicillin, so not very surprising. But what we do see is that when you put the susceptible strain in the presence of our resistant strain, it really rescues the growth of the susceptible strain, so it completely bounces back. So it's able to sort of be protected by that resistant strain in the presence of ampicillin. So in this simple lab environment, we do see cross-protection occurring. But this is all going to be happening in the context of a gut microbiome. So in a gut microbiome, we obviously have more species. We're just looking at E. coli in that experiment, but a gut microbiome has lots of different species in there, some of which are going to be susceptible and some of which are also going to be producing beta-lactamases. And competition, of course, in the microbiota is going to be really, really intense as well. So we wanted to test how this might play out in the context of a microbiota. So we established this anaerobic gut microcosm system, and this contained gut slurry, which is made up of an anaerobic peptone wash containing things like bile salts. And what you can do is you can autoclave that, and then you can reintroduce a gut microbiome. So we had a human gut microbiota, and it was composed of 3 different samples that were combined together. So you're able to sort of test how evolution might proceed essentially in the presence versus absence of a complex gut microbiota. So you can imagine this is our gut microbiota here. And what we did was we tagged a focal strain of E. coli and we embedded it within this gut microbiota. And so we're able to follow the effect of intraspecific interactions within our 2 E. coli strains, so our resistant and our susceptible strain. And we were also able to test the effect of the microbial community on E. coli resistance as well. So this is essentially our experimental design. We had E. coli resistant strains in green and susceptible strains in yellow, and we grew them up either as monocultures, so they're grown by themselves, or as co-cultures, so they're grown in the same tube together. This allowed us to test how these interactions within species were able to drive antibiotic resistance. But then we also had a community treatment. So half of our populations were grown in the presence of this human gut microbiota, and half of them are grown in the absence of the human gut microbiota. So this is really testing how these interspecific interactions shaped E. coli resistance. And we performed this experiment in the absence of ampicillin and in the presence of ampicillin as well. And so remember, what we're really looking for here is, first of all, whether we see cross protection occurring. So can our susceptible strain get an advantage in the presence of either the resistant strain or resistant members of the microbiota? And we're also looking for evidence for competitive release. So can our resistant strain actually get a benefit from antibiotic-mediated killing of our susceptible strains? And they might come from the E. coli susceptible strain or susceptible members of the community. So if we first look at the susceptible strain of E. coli, what we're really looking for here is evidence of cross-protection. So can E. coli benefit from conspecific resistance or resistant members of the microbial community? And so up here on the left-hand side, we have the growth of the susceptible strain just by itself. So no ampicillin around, no resistant strain, is just growing happily by itself. If you add an ampicillin, unsurprisingly, the growth is reduced. If you do this in co-culture, so it's in the presence of the producing strain, we still see this reduction in growth in the presence of ampicillin. And if we do the experiment in the presence of the microbiota, and the resistant strain, we still see a reduction in growth. And so what this means is that we don't see any evidence in this system for cross-protection occurring. It doesn't seem to happen in the gut microbiota. So what about resistance E. coli? Do we see evidence for competitive release? So can resistant cells actually benefit from the death of susceptibles in the population? And so here on the left-hand side here, we have resistant strains growing quite happily in the absence and the presence of ampicillin. Of course, they're resistant. But if you grow it then in the presence of a susceptible strain, you can actually see that it gets a slight increase in its growth. And if you grow it in the presence of the microbiota, you see that the growth is significantly increased. And we only see this happening when ampicillin is applied. So when we've no ampicillin around, the presence of the microbiota reduces the growth of our resistant strain, indicating that competition is occurring. But if you add ampicillin, then E. coli is getting an advantage by being in the presence of the microbiota. So this is really good evidence for the competitive release of antibiotic resistance. So for competitive release, one really prominent assumption is that there is competition between E. coli and the resident microbiota. And we could kind of infer that from our previous data set, but we just wanted to sort of use these supernatant experiments to really quantify the degree of competition between E. coli and the microbiota. And so what we were able to do is essentially grow up either the microbiota or the resistant strain by itself. You remove the supernatant and then you grow the resistant strain in the supernatant. And this can tell you the degree of niche overlap, so how much exploitative competition is occurring between the resistant strain and the microbiota. And it can also quantify basically interference competition. So are there any sort of exoproducts or toxins in the media that are killing E. coli? And so we perform this assay in its own supernatant. So how well does the resistant strain grow in its own spent supernatant, which is where you'd expect to see 100% niche overlap? And how well does the resistant strain grow in the microbiota supernatant? And you compare the two. So here I'm showing you the growth of the resistant strain in each supernatant. So we have a control. Obviously, this is just fresh media. And we have the supernatant of our resistant focal strain, so its own supernatant essentially. And this, I know this doesn't look like it's different, but it is. It's just the scale that it's on. So it is growing slightly less well in its own supernatant than the control, as you'd expect. But if you grow it in the presence of the community supernatant, you can see a huge decrease there. So it looks like it's more than exploitative competition going on between the community and the focal strain. You've probably got interference competition going on as well. And so this is a really good indicator that actually when we add antibiotics to the system, it's killing off susceptible members of the community and reducing the degree of competition between the community and the resistant strain, and it's allowing the resistant strain to get an advantage. And we just verified this then using flow cytometry. So this is a really awful image. But essentially what we did was we calculated the total abundance of these different combinations. So we had the microbial community embedded with the resistant strain was embedded in it. And we had the microbial community with the resistant strain and ampicillin. And so essentially any reduction here in growth is because of ampicillin. And so we did this using lots of different combinations. And essentially this is just showing that the abundance of that community is reduced when you add ampicillin, showing that there are some susceptible members in there. And so one sort of take-home message I get from this is that, really considering this in light of how we treat patients with antibiotics, the current paradigm is to really hit patients quite hard with antibiotics so that we eliminate as much of the pathogen as possible. But actually our work and lots of great work on malarial parasites as well show that competitive release is a really strong driving force for resistance evolution. And so one sort of more promising approach might be to hit the pathogen a little bit more slowly so that that competitive release force is not as strong in the community, essentially. So you don't free up that many resources for that resistant strain. You still have some susceptible members in the community that are essentially competing with the resistant strain and preventing it from taking off. So that's really it. I'm going to summarize the second part of the talk. So we found that while in a lab, a really simplified, well-mixed lab environment, cross-protection can occur. Actually, when we put this in a more realistic scenario, competitive release did tend to dominate. And potentially in light of that, we should think about how we use antibiotics. And if you're interested, there's lots of great research really considering how competitive release drives sort of could predict how we use antibiotics. So Andrew Reid is really good. He's done lots of stuff on this as well. And so I'll finish there. I'll just like to acknowledge lots of people who've been sort of part of this project for so long. Mike and Angus, who were my supervisors, Steve, who has done lots of genomic work with us, Adela, who's now in Argentina, Joe, and Sive as well. And thanks everyone for listening. Take any questions.
Great, thanks, Siobhán. That was really nice, really nice talk. Thank you so much. And there's some compliments as well in the chat of I think a couple of people had to leave early, but thank you very much for the great talk. We do have time for a couple of questions. Fred Adler, are you still there? Would you like to ask your question? Okay, great, thank you.
Yeah, thanks, Sean. That was really fascinating talk. Love the Pseudomonas part.
Sorry, before you go, could I ask Siobhán to please stop the screen share? So
sure. Yeah, is that okay?
Right.
Yeah.
Go ahead, Fred. Sorry.
Oh, no problem. I can't actually see myself, but that's a good thing. So yeah, loved all the stuff on Pseudomonas. So I was wondering whether there's if you have the power to detect any trends in the hyoscyanin production across patients. That is a function of age or the rest of the microbiome, treatment, or somebody else pointed out about host genetics. Can you see anything?
I think there's other people looking at that. So you do tend to see loss of siderophore production within a patient over time. I don't think it's been correlated to any particular, treatment or anything like that, or microbiota composition or anything like that. I think it seems to be really, really difficult to detect any strong trends of that kind of thing in patients. But I know Sandra Anderson and other people have done some great work looking at sort of the sort of spread of cheating within a patient over time.
Great, thanks.
Yeah.
And do we have other questions? Anyone else have a question?
Yeah, I just kind of wanted to ask something a little bit, if no one else has a question.
Please do. Go ahead. Go ahead.
I'm sorry, Joe. I really loved the presentation. The last part really hit me because I work on malaria and I look at competitive release in Plasmodium falciparum, and my PI comes from the Andrew Reid lab. So I thought it was really cool when you mentioned all that. I was wondering, for future studies, now that you found there's competitive release in there, are you going to try to hone in on specific drug doses that can mitigate competitive release?
I think that would be really, really interesting. And I think that there's probably some really nice interaction there between how long the drug stays in the system and whether or not the non-producers can actually, there's probably a really fine line between can the non-producers actually benefit from the susceptible cells actually benefit from the resistance before they're killed. You know what I mean? Or even, I think there's definitely experiments to be done there. Unfortunately, I have left Zurich and Alex's amazing setup there for the anaerobic microcosms, and this was just sort of a one-off project, but I think it would be really cool to explore the interaction between competitive release and cross protection a little bit more. This was a really crude kind of one environment, and it would be nice to sort of change that environment a little bit to see if you can kind of swing it in one way versus another. And then also, as you say, to try and manipulate the dosage and the timing of antibiotic administration in order to kind of constrain competitive release. So play around with it a little bit more. It'd be really cool to do that.
Yeah.
Thanks. That's a great question. I was wondering similar kinds of things about next steps and next directions. Do you have any others that you're interested in pursuing, Siobhán, especially in relation to, actually applying this to human health and to...
Yeah, I think it would be, I think there's so many things there that I'd love to do. I think the cystic fibrosis stuff to me is really interesting because it's very hard to kind of understand how interspecific interactions are driving that. So it's so hard to get to ask these questions sort of in that space between going out and looking at clinical data and then doing these experiments in test tubes where it's really well controlled. And then actually the sort of relevance of that to the real world is so questionable that trying to do something in the middle is really hard, because I think we did it with the gut microbiota stuff, but there's obviously so many outstanding questions, like what are these species being killed? How, we're thinking about really specific interactions, like what is going on? It's so complicated and so hard to try and pick apart. But I'd love to kind of ask these questions in the context of CF. I think that would be really cool.
Excellent. Okay, we're sadly at the top of the hour. Everyone now knows about your work and can reach out to you. Please, everyone, do that and contribute, follow up with questions. I'm sure that Siobhán would love to hear from you and to connect with you.